ANOVA stands for Analysis of Variance. It's a statistical method used to analyze and compare the means of 3 or more groups to determine if there are significant differences between them. ANOVA assesses whether the variability between group means is greater than the variability within the groups. There are different types of ANOVA tests, such as one-way ANOVA, which compares the means of a single factor across multiple groups, and two-way ANOVA, which examines the effects of two factors on a response variable. In essence, ANOVA helps researchers determine if there are significant differences in means between groups, providing insights into the factors that might be influencing the observed outcomes. Here's a step-by-step guide to performing an ANOVA test. Formulate hypotheses. Start by stating your null hypothesis, H0, and alternative hypothesis, H1. The null hypothesis typically assumes that there are no significant differences between the group means, while the alternative hypothesis suggests there are differences. Collect data. Gather data from 3 or more independent groups. Ensure that the data is continuous and normally distributed. Calculate group means. Calculate the mean value for each group in your data set. Calculate overall mean. Find the overall mean of all data points combined. Calculate sum of squares, SS. Calculate the sum of squares between groups, SSB, by summing the squared differences between each group mean and the overall mean, weighted by the number of observations in each group. Calculate the sum of squares within groups, SSW, by summing the squared differences between each data point and its group mean. Calculate degrees of freedom, DF. Degrees of freedom between groups, DFB, is equal to number of groups, 1. Degrees of freedom within groups, DFW, is equal to total number of observations, number of groups. Calculate mean squares, MS. Mean square between groups, MSB, is equal to SSB DFB. Mean square within groups, MSW, is equal to SSW DFW. Calculate F statistic. Calculate the F value by dividing MSB by MSW. F is equal to MSB MSW. Determine critical value. Look up the critical F value from an F distribution table using your chosen significance level, example alpha is equal to 0.05, and degrees of freedom. Compare F value and critical value. If the calculated F value is greater than the critical value, reject the null hypothesis and conclude that there are significant differences between group means. If the calculated F value is less than or equal to the critical value, fail to reject the null hypothesis. Interpret results. Based on your conclusion, interpret the results in the context of your study and discuss the implications of any significant differences found. This process outlines the general steps for conducting an ANOVA test. However, depending on the software or statistical tool you are using, some steps may be automated, making the analysis more straightforward. ANOVA, analysis of variance, is widely used in various fields for different purposes. Here are some common applications. Experimental research. ANOVA is often used in experimental research to compare the means of 3 or more groups. For example, in a pharmaceutical study, ANOVA can be used to analyze the effectiveness of different drug treatments on patients. Quality control. In manufacturing and industry, ANOVA is used to analyze variations in product quality among multiple production lines or batches. It helps identify if there are significant differences in quality due to different factors such as machinery settings or raw materials. Education and psychology. ANOVA is used in educational and psychological research to compare the performance of students or participants across multiple groups. For instance, it can be used to analyze the effectiveness of different teaching methods or interventions. Market research. In marketing and consumer studies, ANOVA helps analyze the impact of different variables, example price, packaging, promotion, on consumer preferences and purchasing behavior across different market segments or demographic groups. Agricultural studies. ANOVA is applied in agricultural research to compare the yields or growth rates of crops under different soil conditions, irrigation methods, or fertilizer treatments. Environmental studies. ANOVA is used to analyze environmental data, such as comparing pollutant levels across different locations or assessing the effects of different interventions on ecological systems. Healthcare and clinical trials. ANOVA is utilized in healthcare research and clinical trials to compare outcomes among multiple treatment groups or patient populations. It helps determine the efficacy of treatments and identify factors influencing health outcomes. Social sciences. ANOVA is used in social science research to analyze survey data and compare responses among different groups based on demographics, geographic regions, or other variables of interest. Overall, ANOVA is a versatile statistical tool that allows researchers to compare means across multiple groups, identify significant differences, and draw conclusions about the factors influencing the observed variations.